So I'm going to start with a quick note, of course, on potential competing or conflicts. As you all know, I'm an employee of SomaLogic. Claudia is at the MRC unit at University of Cambridge, but we have given them a grant. And Peter is at UCSF, but he's also on our medical advisory board, but we don't give him any money. So today we're going to talk about this thing that we call the liquid health check, where we use encoded patterns in plasma proteins to as indicators of health. But first, I want you to stare at this picture, because what we're going to do to start with is to think about what are the other sources of information, obviously other than proteins. And the reason I'm going to ask you to stare at this picture for a little while is because we're going to ask you to vote. In that, if you had to imagine in 5 to 10 years, you could only have one of these things, which one would it be? So I'm going to give you a few seconds to think about that. And then Jared's going to bring up a poll where you will be asked to click on just one of those things. You'll see all of the options. And I'm seeing it now, but you just click on one thing, and I'm going to give you about 20 seconds to choose the one that you want, and you don't have to favor me or proteins. I just want you to be honest about what you think today. And Jared will give us all the results of the poll at the end of our presentations. Okay, I think that's enough time for you all to think. And so, as I said, we'll come back to the results at the end. Of course, my job is to make the case to you that proteins could be that dominant source of information. And this slide is really the setup for that, that DNA is like the hard drive storage of information, but it's no use if you can't get it out. And how that information comes out, and how the effects of genetics in the environment and behaviors and medications and aging, how all those things are mediated is via proteins. And as I'll remind you in a few slides' time, thousands of proteins have actually evolved to be an information transmission system. Their purpose is to transmit information from one biological system to another, or from one organ to another, in a complex networked way. In fact, I like to assert that proteins are like the internet. Yeah, maybe DNA is the storage, but actually the transmission of information and photons and electrons, that's what proteins are doing. They're giving us the information that we seek. Maybe even more convincing is that that proteins mediate the effects of all known drugs. Even gene therapy, even silencing RNA therapies, as well as all small molecules, they mediate their effects by changing the level or the activity or interactions between proteins. So here's the weird thing. Proteins have evolved to be an information transmission system. And when we want to put new information into the body through drugs, we depend on proteins. But we haven't thought of proteins generally as that information detection system. Why is that? Well, this is why. Because we're trying to interrogate the internet with a telegraph. And these dots and dashes are like the usual lab results you get from your physician. If you don't know Morse code, you can't understand them, and there aren't very many dots and dashes. So it's kind of not very comprehensive, it's not multidimensional, and it needs an expert to interpret it. So that's why proteins have been overshadowed by other technologies like genetics and imaging and wearables. So now I'm going to tell you about what we think is the better technology. And the picture you're looking at right now is of a modified aptamer, which is that structure that you see, and binding to a protein. The special thing here is that what we want to do is to convert a protein counting problem, which is difficult, into a DNA counting problem, which is easy. And what you're looking at here is this DNA aptamer, this piece of DNA itself, a single strand of DNA, which when you have a single strand of DNA and you don't have its corresponding partner, it curls up on itself and it binds to itself and it makes a shape with charge. And if you decorate that DNA with those side chains that you see at the top, you can make its shape and charge even more specific, so that you can find aptamers that have an exquisite match to an epitope on the protein. And the way you convert the counting problem from one to the other, I'm looking at this picture. How many protein molecules are there? 1. How many DNA aptamers are there? 1. So if I count the aptamers, I'm actually counting the proteins. So within the SomaScan assay, which I'm not going to go into in detail today, that's the point. How do we create these one-to-one complexes of aptamers to proteins such that we can end up counting the DNA, which is much easier, as you'll see in this little modification, that we can count the DNA on a hybridization slide where we've just simply printed the corresponding sequences of each reagent on the slide. So the assay itself then, it has to enable the binding of the right reagents to the right proteins, which we've partly done by these very, very high affinity reagents that are decorated and modified. But we also capitalize on a second property of the aptamers, which we've selected them not just for high affinity, but for slow off rates. And during the assay, we capitalize on that second component to get extra specificity, because the intended complexes, like the one you see here, have very slow off rates. Often it's hours. So during the assay, there's hardly any dissociation. But for reagents that transiently bind to the wrong protein, those dissociation rates are very short. And the nice thing about aptamers is that they're polyanions. So what we can do during the assay is to dump a lot of cold, unlabeled polyanion into the solution so that it competes with the reagents, which are all labeled. And so those transiently, non-specifically bound reagents that fall off get predominantly displaced by the polyanion competitor. So that's the second element of specificity that we get from the assay. The first being affinity, the second being this kinetic challenge. And through a series of binding steps, we end up with these one-to-one complexes, which we then dissociate, and we count the DNA on the hybridization slide, as I mentioned a moment ago. How do we make these reagents, you might think? Well, if you start in the middle of this slide at the bottom, this is an industrial process where we start with a new protein that we want a reagent to in the bottom middle. And we incubate it with a library. We have several different libraries with different modifications. Remember in the last slide, I showed you that we decorated the DNA with different side chains that look like amino acids. Each library only has one of those modifications in it. So we'll start with one of the libraries and a protein target. We incubate them together. And we find the protein, the reagents, the subset of that 10 to the 15th library that binds well to the target protein and has a slow off rate. So remember, we're doing the affinity and the off rate. In the top left-hand corner, we throw away the ones that don't work so well. We take the ones at the fraction of the 10 to the 16th that bound well and had slow off rates. We amplify them back up again, and then we do it again. And each round of that selects process enriches that library again and again and again for the reagents with the highest affinity and the slowest off rate. And in the end, we sequence the ones that looked the best, and we choose one. Often there are families of sequences which very closely resemble one another, but we choose one to go in the SomaScan assay. And once we've found it, once we know what its sequence is, of course, we can make it by the bucket load for the next 100 years. We don't need any animals or any expression system. They're just oligonucleotides. So this slide tells you the key features of the SomaScan assay itself. As you see at the moment, it's roughly It used to be 1,130, and now it's roughly 5,000 reagents. I think the thing that we're most proud of then is this combination of the precision, the CVs, the 6% CV. That isn't just last week's run. We actually measured the CVs over hundreds of thousands of samples, and that's the average over roughly 150,000 samples. We monitor the CVs very carefully. And that combination of a low CV with a large number of measurements is very important, because a lot of the changes in the proteins that we've found are quite subtle. They can be 5% or 10%. And so you might have other technologies that have an apparently decent sized menu, but if you don't have the degree of precision to go with it, you're not really actually able to measure these physiologically important changes of 5 to 10%. So that combination of precision and number is, we believe, unsurpassed with any other large-scale measurement technique. Even those measuring tens or hundreds of proteins can't match those CVs. So this is, and just finishing up again then, before I open up to Claudia and then Peter, what are we looking for in the blood? Well, as you see on the left, we're looking for proteins that may not be purposely secreted. They may be downstream to cell death or leakage from damage. And then on the right-hand side, of course, there are thousands of proteins which have evolved to be information transmission signatures. So the key point here is that we are asserting that protein patterns encode for lots of useful information, and they've been neglected in the past because the technology hasn't existed. And what you're going to hear about in the rest of the seminar today is where we put that theorizing and wishful thinking to the test, where we designed some comprehensive studies that would actually find out to what extent the protein patterns really encode for current health states, future risks, and the impact of behaviors. And so with that, I'm going to hand over to Claudia Langenberg.
Well, thank you, Steve. It's my pleasure to present our collaborative paper, which we published a few months ago. And this really was an example of a successful partnership between a commercial partner and 9 academic institutions in the UK, US, and Norway that we present here. Now, let's take a step back. So why did we do this? The global burden of multimorbidity requires new strategies for primary and secondary prevention. And disease-specific approaches for the prediction and prevention have been developed and implemented, for example, for type 2 diabetes or the health check in the UK, health checks in the UK. Now, since these are applied to large population groups, the emphasis has to be on cost effectiveness and scalability. But there's also clear evidence of the potential of proteomics to tailor those prevention strategies more to the individual. But this is based on small proof of concept studies that generally focus on single diseases. Now, the question really is whether it is possible to develop what I would call a highly personalized single point test that is scalable. And let me just explain, it's not just a single point, but a single domain test, i.e. we're only looking at proteins, in contrast to predictive models that include many and very diverse parameters. The test is also dynamic or responsive, so it has the potential to monitor changes in risk. And finally, we want to test the application not to one, but simultaneously across multiple health states or diseases to test specificity. And just in terms of background, so my own research and that of the MRC unit at the University of Cambridge focuses on type 2 diabetes, so I will present diabetes-related examples. And why does this matter? It's extremely common, it's serious, it is rapidly getting worse, and it's a large contributor to multimorbidity. And this IDF map shows you the massive global problem. Most importantly, diabetes is preventable. And you all know that the global increase caused by changes in obesity, that it is caused by changes in obesity. And actually, what this means is that we need universal changes that target the obesogenic environment, i.e. shift the whole population, or at least stop the shift to the right. However, and this brings me to our paper, we also need better strategies to identify individuals at sufficiently high risk for us to intervene and prevent it from developing. So for this work, we brought together 5 different cohorts with over 16,000 participants from the UK, US, and Norway. So you can those here. And it was a massive effort to bring these data from the different studies together. And they were chosen and included because they have complementary strength and relevant outcomes of interest. And as you see, so they included diverse populations in terms of their age and sex and their ethnic profile, and that was deliberate. And as you've heard from Steve, so the SomaLogic version 4 platform assesses around 5,000 protein targets per participants. So this work included over 85 million protein measures. Now, we want to know whether this snapshot of the circulating proteome can predict health behaviors, current health states, and future health risks. So these are the measures that we included in the study. So self-reported excess alcohol consumption, objective measures of physical activity expenditure based on combined heart rate and movement sensing. So this is a measure that's not often available in studies because it needs individual calibration. So it's a lot of intense work. Then self-reported smoking, and in terms of current health states, ultrasound-based assessment of liver fat, creatinine-based estimation of kidney function, and then DEXA measures of body fat and composition. So DEXA is a method that uses low-energy X-ray beams that can measure the percentage of your fat, your lean muscle, bone density, and then ultimately exercise tests with measures of maximal oxygen uptake, which is a measure of aerobic endurance. And then future health risks, here we focus on the conversion of pre-diabetes to diabetes and the probability of a first cardiovascular event within 5 years. So I've already introduced here the studies we included and the measures that we model. And I just want to say 1 or 2 sentences just about the modeling strategy. So we use different supervised machine learning algorithms. And actually, to know all proteins were protein sets that were pruned after univariate analyses. And as standard for these methods, the studies were split into 1 to 2 training sets, so here training refinement, and a separate independent validation set depending on the study size. And we set the bar high, so model success was defined as protein only, i.e. only considering proteins in the model performance equivalent to the best available comparator. So that means a high correlation with what we call the standard or the truth standards, let's say the DEXA, so a correlation of 0.6 on R squared greater than 0.36, or an area under the receive operating curve of greater than 0.7. And I'm going to start at the end. It's 11 of the 13 models we considered were successful. As you can see, and included a total of 891 unique proteins, and an average of 12% was shared across these different models. So for metabolic health, let's first consider our plasma protein models for body fat and composition against the DEXA standard that you can see here. And the high R-square values here for these different examples, in other words, the very tight correlations between the protein models and the measures is obvious. And this is not just for lean body mass, total body fat, but also visceral fat, which is a metabolically unhealthy way of storing fat compared to other compartments, such as the hips or the legs. And for health behaviors here, as I've already said, so for most studies, physical activity is only self-reported. And here we compare our protein model against the objective assessment of energy expenditure and achieve an R-square of 0.38. And that is pretty amazing in my experience. And we also see good performance, so an AUC of 0.82 for excess alcohol consumption. And this is the model in women, but actually it looked very similar in men. Now, in the general population, and you will all know, this is the best strategy to identify people at highest risk of developing diabetes is obviously to find those with pre-diabetes, i.e. glucose levels that are just below the diabetic or a diagnostic threshold. But here we consider the risk of people with existing pre-diabetes to progress. So if you look at the y-axis here, this is the proportion of people without diabetes over time, and the extremely low proportion of people classified as high risk by the proteomic model who remain free from diabetes after 10 years is evident. And then allow me a small excursion to some unpublished work. And this is about the specific form of prediabetes or impaired glucose tolerance. So this is diagnosed through an oral glucose tolerance test, a standard dose of glucose. We measure the glucose levels at 2 hours, and if this is elevated but below the diabetic threshold here, or this is here in old money, this is 140 to 200 milligrams per deciliter, if you're more familiar with that, then that's impaired glucose tolerance. And it is underappreciated. It's extremely common, and particularly at older ages, and is strongly predictive of cardiometabolic diseases. It's very rarely measured now in clinical practice because of the complexity, logistics, the time and the cost, because you have to wait for 2 hours, second sample, and so on. And people also don't really like to drink. In the UK, we often give them Lucozade. People don't really appreciate that that's the standard dose of glucose. But so if this is isolated, so isolated impaired glucose tolerance, that means it is missed by the tests for diabetes that are now recommended, i.e. fasting glucose and HbA1c. So you can imagine if you have this isolated, you're not going to be picked up, so you're at high risk of diabetes, and it is very possible that the first time these patients present, it's already with the complications and, for example, not loss of vision, but impaired vision or other cardiometabolic consequences of their impaired glucose tolerance. So the question really is, can we use the fasting, or develop a fasting proteomic signature, so a very simple test to predict this isolated impaired glucose tolerance? And the Finland study is one of the studies included in the previous work. We did the standard oral glucose tolerance test. And so we developed a model using very similar methods that I've previously mentioned, with the training and optimization set and independent validation set. And in the interest of time, I'm just going to talk about the isolated IGT, and which you see here, the curves, we compared lots of different models. And the bottom line really is a 77 protein model here, which you see here, alone performs better than any other non-invasive or invasive prediction model, including fasting plasma glucose or HbA1c. And that's not improved here, considering other risk factors. And in contrast, any of the clinical models is improved substantially by considering the 3 top proteins only. Now let's look at the top 10 selected proteins in the model for either IGT or isolated IGT. Now, in Finland, we have genetic information on our participants, and we can classify them based on high or low genetic risk for different metabolic parameters. And that's what you see here in this panel. So here you have, let's say, high genetic susceptibility to high insulin levels, insulin resistance, increased risk of diabetes, or body mass index or obesity. Now, the significant associations that you see here in this panel between genetic risk and the proteins show that the selected proteins are not only good predictors, as I've shown you previously, but reflect the underlying genetic etiology. And so here in the last panel, you see this is in the independent Whitehall-2 cohort. So that's also part of the paper we're presenting. They were also associated with a future risk of developing diabetes in the expected direction, so consistent with what we've previously seen. And my conclusion here is that the identified proteins are good predictors of the current health state, so isolated impaired glucose tolerance. They reflect the genetic risk and etiology, and they associate with future disease risk. And so with this, I would like to hand over to Peter, and thank you all very much for your attention, even though it's not really in person. Thank you.
So good morning, everyone. I will transition now to discussing cardiovascular risk modeling through proteomic insights that we've obtained from several studies. Just to recapitulate what you already likely know, cardiovascular risk models are very important because we use them to guide therapies. So our current cholesterol or lipid management guidelines are based on risk assessment to which we tailor the intensity of lipid management. Similarly, our current blood pressure management guidelines, hypertension guidelines, also rely on risk assessment to determine the intensity of treatment. So it is really important for these risk models to perform optimally. Unfortunately, the currently favored or used model, at least in the United States, the 2013 American College of Cardiology, American Heart Association pooled cohort equation, overestimates the risk in healthy population by about a factor of 2. So we refer to this as poor calibration, but the implication is that several million people are treated unnecessarily with statin therapy because their risk may be overestimated. Conversely, there are other populations, some special populations where the pooled cohort equation underestimates the patient's risk. Examples being patients with chronic kidney disease or patients who are infected with HIV. Also, on an individual patient level, we know that the pooled cohort equation misestimates the risk. If we look at some metrics of discrimination, which tells us who is likely to have a disease and who is not, we usually rely on measures like C statistics or receiver operating curves. And based on those criteria, C statistic is typically less than 0.7 for the pooled cohort equation. You know, again, speaking about less than optimal discrimination with room for improvement. And lastly, traditional risk factors perform particularly poorly in patients who already have established cardiovascular disease. And one reason for that is that the patients who are the sickest tend to have the best risk factor profile because we treat them most intensively. So you could look at someone who has a great risk factor profile who actually will have a poor outcome because they are the sickest patients. So risk models that rely on traditional risk factors can be a little bit misleading in patients with established cardiovascular disease for this reason. So our work, this was collaborative work with SomaLogic and UCSF and a group in Norway, the Cohort. We focused initially in the cardiovascular arena on developing and validating a protein-based risk score for cardiovascular outcomes among patients who already had cardiovascular disease. So these patients had stable coronary heart disease. And the dogma was there was no reason to risk stratify these patients. They already have disease. They should all be treated intensively without risk stratification. It just wasn't felt to be necessarily useful. But we looked into it with proteomics and we decided to develop a risk model for patients with stable coronary heart disease, where the outcomes would be the future risk of myocardial infarction, stroke, heart failure, or death. And we had a discovery cohort where we developed the protein risk score with a time horizon of approximately 4 years. This was a cohort based in San Francisco called Heart and Soul, 938 participants. And we measured 1,130 proteins in each of these participants and applied statistical techniques to find proteins that were statistically prognostic of this composite outcome. And a little bit to my surprise, we actually found more proteins than I expected. About 200 proteins were statistically prognostic of cardiovascular outcomes. And then we use machine learning approaches. One of these is called Lasso, to create a 16 protein risk model that predicted the 4-year risk of that cardiovascular outcomes. And we made the model a little bit smaller by eliminating the least important proteins and ended up with a 9 protein risk model. And we took that risk model into a completely different cohort to validate it. This was a cohort in Norway called HUNT-3. So this is now the one slide that I want to show from this study. On the left side are data from the derivation cohort, that's hard and soul, the 9-protein model. On the right-hand side is the Norwegian cohort, the hand-free cohort. And what's shown on the x-axis are in each cohort are the deciles of predicted risk. And what's shown on the y-axis is the four-year rates of events. And as you can see in both cohorts, the predicted risk, shown in the pink color, very well matches the observed risk. So we call this, good calibration. But as a clinician, I'm a practicing cardiologist. What really struck me, what was an eye-opener to me, is that stable coronary heart disease, which I thought was a fairly homogeneous disease that didn't require risk stratification, really represents a wide spectrum of risk. In decile 1, there are patients where we predicted a 4-year risk of 6 or 7%, which matched the observed risk. But at the other extreme, in decile 10, there are patients whose risk was 10 times as high, really in excess of 60% of having one of these 4 outcomes over the subsequent 4 years. So as a clinician, I then asked the question, why should everybody be treated the same when the risk spectrum is so broad? Now, maybe they could be treated the same if the treatments are inexpensive and free of side effects, like statins or aspirin. But now we have agents that are quite expensive, like PCSK9 inhibitors, that when they came to the market cost about $13,000, $14,000 a year. And even now, they still cost about $5,000 a year. So perhaps not everyone needs to be treated with these expensive agents. And we provide here a means by which patients could be risk stratified. Just one aside, as an interventional cardiologist, I also have to say that one area where interventional cardiology has suffered recently was in interventions for patients with stable coronary heart disease, where stenting probably is no better than optimal medical therapy. So that's taken a lot of business away, and I'm glad it did from our interventional cardiology community. But seeing the data I'm actually showing you, I do wonder whether in the future we might want to examine whether some of these patients that we can identify now as very high risk, in fact, might benefit from an interventional procedure like stenting. So there's no evidence for that yet, but we now have a way to pick out the very high-risk individuals with stable coronary heart disease. So when we started with SomaLogic, when I started my collaboration and we derived this 9-protein model, I was very glad we did, but at the same time, I realized that having 9 proteins measured by a platform that in fact was capable of measuring 5,000 proteins did not take full advantage of the platform. And perhaps the platform could offer much more than some cardiovascular insights. And perhaps it would be a means to examine the health of the entire human body from head to toe. And so with SomaLogic and with some of the academic collaborators like myself, begin to develop this model of using proteomics to inform the health of the entire body, initially by gathering insights about risk of diseases and tailored treatments, no matter where the disease is in the body, storing this knowledge in what was called the health warehouse, and ultimately use this extensive knowledge that will be accumulated in tens and tens of thousands of patients to develop validated health applications that predict risk of diseases throughout the body. So that really was the goal that Claudia so beautifully outlined, and Steve. the goal was really to develop risk models for more than 100 measures of current health, modifiable behaviors, and incident disease risk with a single measurement. So Claudia already beautifully covered some of the outcomes that we described in the Nature Medicine papers. So we had 11 health outcomes that we successfully described. In the left-hand panel, we are showing the cardiovascular outcome. Here it's shown simply as a survival curve, or what we call the Kaplan-Meier curve, where the patients were divided into bins of risk. The blue line is the reference risk, and the other lines shown the progressive increase in cardiovascular risk, so that the bottom line actually shows patients where we can predict the risk to be 6 times or greater than the reference group. So we again see the ability of proteomics to predict and then to observe risks that show a broad spectrum, of what we would call a broad dynamic range of risk. So again, this will be clinically highly useful. So what are some of the current research directions to keep building on what I've shown you? Well, one thing we want to build into these risk models is the element of time. So I've shown you what prediction at one point in time can do to predict cardiovascular risk. But the risk we know changes in patients. So we are actually now beginning to demonstrate that the risk models that we develop change over time. So using proteins, we can show that patients' risk will change over time as the risk changes. So proteins are capable of demonstrating risk as it varies over time. And we also want to be able to show that the proteins are capable of capturing the effects of treatments. And so we have actually looked at several treatments now. Some haven't made it to the market because it turns out that they were hurting patients. There was a drug called torcetrapib that had to be stopped from development. But we actually did a proteomic analysis on it, and we were able to show that the proteins would have predicted the harm of this drug even before it was clinically observed. We showed conversely that atorvastatin, we could capture the benefit of atorvastatin using proteins. So again, a favorable treatment, we could actually predict that it would be favorable based on the changes in the proteome. And similarly with other drugs like GLP-1 agonists that are being used for treating diabetes. So I think what I've shown you so far is that we can use proteins to build risk models, and if we could do it for multiple diseases at once, it would be greatly more convenient for patients to have a single blood test and multiple visits to doctors to have be able to understand their tests. So there's a lot of convenience for a single blood test to tell the entire story about the human body. But there are also some potential cost advantages. And this was part of the Nature Medicine publications, where for the 11 insights, health insights that we derived, we estimated the cost in the British healthcare system. The right column shows the references. And this cost would have been over 2,000 British pounds, I suspect quite a bit higher in the US system. And although I can't really quote you a price for the SomaScan today, I don't actually work for SomaLogic, we estimate that the expected SomaScan costs are a fraction, maybe 1/10 of the cost that would be accrued through the traditional tests. So I think there may be both a patient convenience advantage and a cost advantage. So I just want to finish with three or four more slides before so I just want to reiterate the concept that we start with the blood sample, do an assay, which allows us to learn of current health and risk of diseases. Now, some of you may say, aha, haven't we heard this before? Wasn't there a company in California that in some way had a similar idea, but we know the disrepute in which they fell. So I want to assure you that SomaLogic and the academic collaborators are the exact opposite. We believe in complete transparency and openness. We publish all of our data in great detail in top peer-reviewed journals. Currently, there are about 250 publications based on a SomaScan. At least the publications I've been involved in contain extensive supplemental data that are typically much longer than what academic institutions publish, again, because we believe that we need to be completely transparent. And for NIH-funded cohorts that SomaLogic collaborates with, SomaLogic is committed to public data sharing. So I think this is really the way to behave. So my last two slides, I don't work for SomaLogic, but the usual first question I get when I speak to audiences is any of this available for clinical use? And I would say there is kind of an early version of this test based on the Liquid Health Nature Medicine paper. I think this shows about 9 of the insights. I think I would probably call this somewhat of a limited release, but again, you may wish to speak to SomaLogic because I don't work for them. And lastly, I think what we showed you is really just the beginning. There is far more to come. What I'm showing are the tests that we expect to add in the SomaLogic platform in 2021 and 2022 and beyond. I'm really excited about many of these new models. Models for non-alcoholic fatty liver disease. There are models for Alzheimer's disease. I've actually seen a very useful model for Alzheimer's that is being developed by one of the universities. So you can kind of peruse this list. We've developed a model that predicts progression of chronic kidney disease at UCSF. So again, there's far more to come than what we've been able to show you. So I'm going to stop here and turn it back over to the moderator.
Great, thank you, Steve, Claudia, and Peter. As Steve spoke about it at the beginning of the call, a poll, and we wanted to revisit this slide, thinking about the dominant sources of new medical information in the next 5 or 10 years, and once again provide a new poll for the next 20 seconds or so, to again select one thing that you think is the most dominant new medical information, or the source of medical information. And I'll give you time to do that. All right, looks like we're having a slowdown. So I will end that one. And what I want to show you is just the results between these two polls. So what you're seeing is on the left. The left is the poll at the beginning of the of the talk, during Steve's talk, and then on the right is at the end here. And what we're looking at is proteomics, and definitely proteomics won in both instances, but it looks like the percentage has definitely increased over the 40 minutes of this talk. Steve, Claudia, Peter, do you guys want to comment on on that at all, the difference?
It's Steve here. So I think it's interesting that there's already on the first poll, there's already a selection bias actually for people who on average liked proteomics more than average. When I've done these polls before in big meetings, it's usually less than 10% of the people think proteomics up front. So you're already a biased audience, but it's gratifying to see that that some of you were swayed by what we said as well. I said to the others at the beginning, it's a bit like when you get your car serviced and they come to you at the end and say, oh, please give us a 5 out of 5 or a 10 out of 10, otherwise we won't get our bonuses. Nothing like that is dependent on this. We're just curious about the nature of our messaging, so thank you for responding.
Yeah, and Peter Ganz here, I would concur with Steve, but I would also say, I think we have a lot of smart people in the audience.
Great. All right. So now I've just unmuted all of our panelists, and we'll answer some questions. And this first one, there's actually a couple of questions about the the 9 protein test for Peter. Has the 9 protein risk score been prospectively validated?
Yes. So the 9 protein risk score, I would say, is not the final cardiovascular risk score. So we are open to modifying it, and it is being prospectively validated now in several other cohorts that I think in total encompass another 17,000 patients or so, if I remember correctly. And these cohorts, this more of a prospective look now to validate proteomic cardiovascular risk score, are important because they look across different racial groups. So, which is important, I think. They look at whites, and they look at blacks, and they look at Asians in Japan. They look at the ability of these risk scores, as I mentioned, to change over time. So that's another form of validating the veracity of the score. And again, in some of these studies, they also look at the ability of these scores to capture changes with drugs. Now, none of these are prospectively planned studies where there is typically some blood sample that's frozen. So we are looking at blood samples that were stored, in a sense, retrospectively. But all the ideas for testing hypotheses are really, these are prospective hypotheses. So the risk score may not end up being a 9 protein risk score. It may grow a little bit in size, but there will be much, much more additional information coming out, which I would call very solid validation.
Okay. And forgive me, I think you might have mentioned that age is being looked at as well, because that was another thing that someone was asking
Correct.
about.
So one of these cohorts, in fact, looks at how well the risk score performs in elderly patients. So age will be making sure that the risk score works not just in kind of middle-aged people, but also in the elderly is part of the validation. I can tell you right now that it works well in the elderly.
Great, thank you. I think this question is for Steve. Someone writes, I'm curious about the ultimate application for this platform. The notion of a liquid health check points towards a possible set of applications, but could the panelists expand on this? Would the focus be on consumers or healthcare systems?
Thanks. That's an interesting question. And the first set of 10 tests were released until the virus hit in integrative physicians and concierge physicians who are primarily interested in preventative health. So they're not treating chronic disease. They're not specialist cardiologists. They're people who are treating and who are in the business of prevention. So there's a group of tests and many of the things that you heard Claudia talk about are useful for that purpose. And then in the pipeline that you saw Peter talk about, there are a lot of tests there that are much more relevant to chronic disease management, heart failure prognosis, kidney function decline, and the secondary cardiovascular test. And those are much more geared at health systems. But in the end, as we engineer out the cost and as we qualify non-invasive matrix like urine, we think that a lot of these tests will be attractive to consumers and we'll be able to do home collection and we'll be able to get many of these tests ourselves. So those are the three markets that we're interested in. How exactly you will choose when there's a pipeline or there's an available menu of 100 or 150 different tests, we're flexible on. At the moment, we started out with saying everybody gets all of those 10 tests. You don't get to choose. But as we evolve, it's going to be more of a menu-driven thing. There may be groups of tests. For example, Peter talked about choosing the right drug for residual cardiovascular risk. There are drugs, SGLT2 inhibitors are particularly good at people whose kidneys are vulnerable and who are more susceptible to heart failure than other risks. So if you combine the residual CV risk with heart failure and kidney, you could allocate people to SGLT2 inhibitors. And similarly, GLP-1 agonists are good for diabetics and cardiovascular risk reduction in the diabetic population who maybe don't need kidney protection. So once again, if you had the right group of tests, you could imagine matching a set of 6 or 7 different tests to a population that had unresolved cardiovascular risk that would enable you to choose intelligently between the different available marketed drugs. So I think that we're going to be flexible on how these are grouped and how they're chosen. And the platform enables a great deal of flexibility over how that will be accomplished. And we'll have to learn, as we collaborate with health systems, have to learn how best to do that. But the platform doesn't constrain us because we always measure all 5,000 proteins.
Great. And then I think also for Steve, what are the variances among different ethnic groups and diets? Actually, that one might be for Peter. But if either of you guys have a comment on that one.
Or maybe even Claudia, variances in what types of diets various ethnic groups consume and how it's reflected on the proteome. Is that the question? Which sounds very reasonable.
Yeah.
Steve has, because I'm actually not aware of looking at the effects of different diets among different ethnic groups. Steve, does SomaLogic have any kind of information to address that? I'm not aware of it myself.
Well, to the extent that ethnic groups are represented in the big cohorts that we use, any useful information will already be incorporated in the models. But I know Claudia has some specific information on the Mediterranean diet and the direct diet in her Finland population that she might want to talk about.
Yes, it's just briefly to add. So there are efforts ongoing and not published yet, but to kind of quantify and describe the proteomic associations for a variety of kind of or selected dietary scores, I would say, or different components of diet. I would say for what we've discussed today is I think that's what Steve was alluding to. I think the beauty of this and the protein only models is that, sure, we would expect that some of the power of the prediction models is because some of the proteins reflect some of the behaviors, as we've shown for some, not diet, and hence that's what contributes to the associations for diseases that have a dietary component. So that would be my answer.
Great, thanks. We have a number of questions on just the regulatory strategies for bringing this test on. Does anyone want to comment on just the strategies that are out there that would be best suited for this?
Yeah, I can do a quick one on that. So the 10 tests we released already are released in the United States as CAP clear qualified tests under as laboratory developed tests. We had our first conversation with the FDA last week about about qualifying the platform itself and taking the particularly the high risk tests through more traditional FDA pathways, and so we're starting on that. And also because the tests are actually software, that if once we've got the platform qualification process agreed with the FDA, the ultimate hope is after doing several more traditional submissions and after the platform itself is accepted as a regulated platform, that each new test is actually regulated as a piece of software, but that's open for discussion. And then finally, some of the tests which are useful in drug development, we also have the option of taking them into the biomarker qualification process at CEDAR, and we've talked to them preliminarily about that too.
Great, thank you. And then Claudia, I think this question would be good for you. What are the pros and cons between proteomics and metabolomics approaches?
Oh yes, so that's an interesting question, and I've been asked this before. I think my short answer would be that they're extremely complementary. We've done a lot of work now on proteomics as well as metabolomics, and I guess it starts with genomics or the genomic data integration. And I think the beauty of these different layers of omics is that if you integrate them and you have the ability to integrate several of these, really they show such stronger associations with diseases because they're more proximal than compared to when we study genetic effects. And so I think, I know it's an indirect question, I think where possible I would always advise it's good to get both. And it depends on the focus, obviously, of the question. Yeah, so a bit sitting on the fence, I'd say it's very complementary.
And Peter,
thank you very much.
Can I just make a comment? Because I have to confess that I was completely unbiased with respect to metabolomics when I started, but I'm part of an NIH effort in the field of chronic kidney disease, which uses both proteomics and metabolomics. And the metabolomic predictors of chronic kidney disease outcomes, whether it's progression or cardiovascular risk, the metabolomic predictors are very few compared to proteomic predictors, and so few that they are actually unable to even build any risk models. So at least in that area, proteomics is far more useful than metabolomics. That's been my general experience in cardiovascular field, that metabolomics actually has had far more limited utility than proteomics. So that's just one person's take on this, but I've been largely disappointed, at least in my area, with metabolomic findings to date.
I think one of the things I could maybe add is, I mean, I guess if you're talking specifically about prediction, that's one thing. The one thing I would note of the studies that we have done on looking at metabolites to diseases in terms of their associations, it's a lot less specific, I would say. And so the associations between metabolites across a whole range of, we looked at over 50, new diseases developing over the follow-up, are very, very shared. And so much less disease specific, and hence, I would say that's a limitation if you're interested in the very specific etiology of a disease or infer about causal pathways.
Correct. And the line from genes to proteins is pretty straightforward. And the lines from genes to metabolites is far more complex.